Hey folks, welcome to lecture 4. I'm going to share my screen and we can get started. We're going to talk a little bit about some more data and hypotheses questions. Just take a listen. There will be a quiz at the end, but remember I am posting these lecture slides as well, so you don't have to memorize everything. It's good to take notes, but the slides will be up as well. So first we're going to talk about exploring your data and which test do we even do. So I've talked to you about how you need to come up with your statistical test prior to collecting your data, but how do we even know which one is right? So today we'll talk about how to distinguish different types of projects, how to formulate a hypothesis as well as a null hypothesis and what that even is, and then how to choose the best test to use. So there's different types of projects, 4 main types. We have a descriptive, differences, correlation and regression, and an association. Descriptive project is where you use a bunch of measures to summarize something instead of testing a hypothesis. So what does that even mean, right? So as an example, if I asked you to go on campus and walk around and write down all the plants, you can't really analyze that using any particular statistical test. So you use it by measuring diversity, and that's a Shannon-Weiner index, and we will learn about that later. In a differences project, you're literally looking at least 2 samples of data and using summary statistics to describe them. So a little bit like what you've been doing, where you find the mean, the median, you make a histogram, you do a little bit of preemptive statistics to look at the samples, and then you test them to see if they're different and if they're different from each other. And a lot of times there's more than just 2 samples. We use this a lot in biology and environmental science. A lot of times are those different kinds of variables which you'll have to add in for your test. So in a correlation and regression, we're looking at linking variables instead of looking at what's different about them. So you're looking at the strength and the direction of link between variables. So what does that mean? All right, I looked at sand crabs for my grad school project, and if they ingested microplastics, did it matter? Like, did it make them slower or faster or cause any changes when they were trying to get away from like a bird, like burrowing down in the sand? Well, a correlation to that is that's been tested already is that the different kinds of grains of sand based on size directly affects and is correlated to how fast they burrow into the sand, right? I also found and reproved from another test that parasites in their little bodies, um, with those parasites, it's a direct correlation showing other issues and other variables. So then we did a regression analysis, which is like an extension of the correlation, where you take all the data and you do a mathematical analysis of it to show the links in those variables. And you can have a multiple regression analysis. You can have like it's crazy how many different kinds of regression analysis there are out there, but that's the concept of it. So the correlation is looking for the direction of link, and then the regression is the mathematical part of the correlation, if that's how you can remember it. Then in an association project, this is when you're looking for a link between the categories of data. So you might be counting things that fall into different categories. So let's see, if we're looking for species, you go to the park near your like closest park to your house and write down all the different species that you see, like tree, tree, tree. So you make little tick marks. Okay, bird, 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 bird. Dog, 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 right? Human, human. So you would count how many different species there are in that study. Does that make sense? Then you would look and see if there's a link. The more dogs there are, the less birds there are. Is that something that happens? The more people there are, the more dogs there are. Those are kinds of different links. So you would be looking for those different links between categories. So then we have to talk about a hypothesis. So some people get scared of this, and that's okay. It kind of baffled me in the beginning too. Like, what the heck is a null hypothesis? So basically, it's a research question, and then you do statistics to either prove that it's true or not. Okay, so it's either going to support or reject your hypothesis. In the null hypothesis, or what we call H0, it always means that there's no difference between samples. And you test that to see if there is or not. And if there is a difference, you reject the null hypothesis. Then you try to prove if your original hypothesis is supported. And hopefully your original hypothesis isn't just there's a big difference. It should be something a little bit more specific or predictive. Um, an example is that the purple sage plants along Water Canyon on the south side, there's going to be way more of them because it receives more sun. Okay. And so then the null hypothesis would be that there's no difference of purple sage. So then, we might find that you have to measure the amount of sun and the purple sage plants, maybe there's a difference in like size and whatnot. So you would go from there. Choosing the correct test. Now I grabbed this from the book that I like a lot. There's a lot of these out there, but I like this one especially for our class because it keeps it pretty simple. I'm not going to give you any kinds of crazy data for this particular class. If you go on to capstone or do an advanced statistics class, then you'll learn some more stuff. But this is it's a pretty basic good one. All right, so you start here. Do you have one single sample that you want to describe or more than one? Well, we usually have more than one, right? So are you looking at the difference between samples or a link? Yes, we are looking at the differences. Or are we looking at maybe correlation? So I was looking at correlation, so I went down here. Do you have categorical data? Do you remember what categorical data is? So categorical data are like categories like cars and then the color of cars, okay? So if you do have categorical data, you go down here and you either have a single or several categories. You go goodness of fit test or chi-squared. If you don't have categorical data, and then you have to test and see if they have normality or not. And that would be like doing the histogram, running you have to run your normality tests. And we did that in our R, we run the Kruskal-Wallis test. Okay. And if it is normal, then you could do your linear regression. If it's not normal, there are what we call non-parametric tests, and that's the Spearman rank correlation tests. So we'll get more into that as we go, but these are step-by-step kinds of things that you want to look at. Here in the middle, samples in matched pairs is something that you would pair together, like in an age group. It's basically something that's a shared characteristic. So if you were looking at, I'm trying to think. So like gender, all the males, all the females would go in one group, all the males in another group. Age, anybody between 10 and 20 age group. I did categories or matched pairs with size, carapace sizes, so the size of their shell. So everybody between 10 and 15 millimeters went in this category, kind of things like that. Okay. And then it looks at different kinds of replicates, how many replicates you have. You always have to test your distributed data, whether it's norm. So if you have numeric data, you have to see if it's normally distributed. That's just something that you can kind of remember. All right. So that's it for today. I'm going to add a couple questions in, and remember, these slides are posted.